Okay, hi folks. This is week 4 lecture review, and I anticipate that a lot of people have not yet viewed all of the lectures on energy, enzymes, membranes, and cellular respiration. Cellular respiration makes up a big chunk of this week's lecture material. But I'm going to go ahead and go through some of those things, and I want to tell you about next week. Next Thursday, let me make sure I'm getting my dates right here. I think next Thursday is the 23rd. Is that correct? 20th? Yes, the 23rd is when your exam 1 is going to open. It will open at 9 a.m. Thursday morning, and you will be able to take exam 1 anytime between 9 a.m. Thursday and 3 p.m. Saturday. So if you work during the weekdays, you could take it Saturday morning if you want, but it gives you some flexibility. I highly recommend that you make a plan and take that exam at the earliest time that is convenient and works for you. Don't leave it to the last minute for no good reason. I've seen things go badly when people think they can just wait until the end and then realize they don't have time to study as much as they needed, or sometimes things go wrong. So make a plan for when you're going to take that exam and prepare to have all of your studying done and get it taken care of before Saturday at 3 p.m. Next week will be a little bit different schedule-wise for me too. I'm actually going to be traveling Thursday afternoon and through the weekend. So I'm going to, instead of doing a lecture wrap up on Thursday next week, I'm going to do one earlier in the week, probably on Tuesday. I'm just going to make myself available for people who have questions about any of the review material that's relevant to the exam. By the way, this exam is going to cover weeks 1, 2, 3, and 4. So this material that we have through cellular respiration is all the material that you need to know for the exam. How do I recommend you study for the exam? I recommend that you get those weekly review questions. So I'm going to go to this week 4 module that's here. And I'm going to open the lecture material. And you can see that there is a lecture on energy and enzymes and membranes, part of that as well. There's also a little enzyme animation, which is very helpful, and I highly recommend that you watch that animation. And then my lecture on cellular respiration and fermentation. You also have the PDF of the slides, which I recommend you access and go back and look at those slides or look at them during the lecture, help you take notes. This is something that you should be doing every week. And this is the best way to prepare for your exams. And specifically, going back to this module, look for those weekly lecture review questions. They have been posted every week this semester. So there were week 1 review questions, week 2, week 3 review questions. Here are the week 4 review questions. Access those questions. As you are going through the lectures and you're taking notes and you're looking at the slides, if you're doing that, think about the answers to these questions and write the answers down somewhere. Some people like to make flashcards. I'll say flashcards sometimes work, sometimes they don't work. You want to make sure that you're going beyond just recognizing something like, okay, yeah, I know what that is. I know what that is. But you want to make sure you get to the point where you can recall it if it's not in front of you. So you answer all these questions where I say, know the following terms: kinetic energy. You should be able to tell me what kinetic energy is. That's the energy of motion. You should know what potential energy is, energy that is stored. You should know that chemical energy is a type of potential energy, and it's specifically the type of potential energy that is stored in the bonds of molecules. So ideally, as you're answering all of these questions and getting all of this information together, then you are talking through it to yourself or to other people that you know. And at some point, to really test yourself to know that you know this stuff in preparation for the exam, maybe you could go back to this original document. I mean, it's possible you've saved a copy for yourself that you can edit, but then go back to the original and see if you can answer the questions without having the answers written down in front of you. That's the ultimate test about whether you know it, or have people ask you things out of the blue. So my recommendation for preparing for this exam is to study these questions on the review sheet. If you know all of these information from weeks 1 through 4 review sheets, you should do well on the exam. Get the answers to these questions from your lecture. That's where they are. Okay. Does anybody who's here want to add anything to that or have any questions about that? I'll reiterate, the answers to these questions are in your lecture material. So don't go to the book or don't go online. I mean, you can look in the book for sure, but maybe if you look in the book, you're going to get distracted by other information. The book is great to help reinforce these concepts, but your first source should be the lectures. Okay. Let's talk about the review questions then. So I'm just going to go to these review questions here that we have, and it starts with energy, enzymes. Does anybody have any specific questions that they want me to answer regarding these? Any specific questions? Okay, the discussion. Oh, I need to go look at that discussion. All right, I'm going to the discussion to see what those questions are. Thanks, Adriana. And I have some too. I want to talk to you all about ATP and ADP and that ATP cycle, which is really important. So let's go to the course Q&A. Oh, gotcha. I'm going to put these in the chat. That wasn't what I needed. Okay, thanks for bearing with me here. We will start answering these questions. Chemical energy versus potential energy is the first question here. So chemical energy is a type of potential energy. And if we look at these different types of energy, we have the kinetic energy, which is the energy of motion. And then potential energy is any type of energy that is stored energy. So potential energy, there's not motion taking place, but there's the potential for that energy to be used. And it could be converted into kinetic energy, right? So the example from lectures, if you ride your bike to the top of a hill, then you are using kinetic energy, which is actually processing food and getting ATP and using ATP to get to the top of that hill. But that at the top of the hill, you have potential energy because of your position or your location, right? All you need to do is let yourself go. And then that potential energy due to the position is going to get converted into kinetic energy of motion. Chemical energy is a specific type of potential energy. And you might notice here that it says potential energy is stored either by location or structure. Chemical energy is a type of potential energy that has the energy stored in structure, and it's specifically in the structure of molecules. So let's look at some molecules here, like this amazing molecule, C6H12O6, which is glucose, which we know is a high energy molecule. This is a monosaccharide. One of the functions of sugars, the primary function, is to provide energy. And that's because this molecule has a lot of potential energy in it. The energy is stored in these bonds. Every bond in this molecule between a carbon and a hydrogen, or a carbon and an oxygen, or a carbon and a carbon, every bond has a specific amount of energy in it. And if you break those bonds, the energy in those bonds can be released. And that is how it is potential energy. And this is very relevant, especially when we're talking about glucose, to cellular respiration, because the whole process of cellular respiration is about breaking apart this glucose molecule and releasing the energy in those bonds, releasing that chemical energy, that potential energy, and using the energy that is released to make ATP. So we can talk about that in a few minutes. That was one question that we had here. And the other question is, ethanol has a lot of potential energy in the form of chemical energy. Yes, exactly. So in lab this week, you may have, you're thinking about all that energy from glucose that didn't end up in ATP when fermentation takes place, because most of that energy is now in ethanol if fermentation is occurring. So yes, ethanol is another high energy molecule. It's got a lot of energy in those bonds, and that is a form of chemical energy, which is a type of potential energy. Does that make sense?
Yes, thank you.
Thanks. So now this other question is, from what source do plants get energy to make ATP? Is that on the review sheet? Let me see what that says. Aha. Well, this is interesting because when we're talking about plants, what is the initial source of energy for plants? Plants initially get energy from where?
Sun.
The sun, right? So they get energy from sunlight, but they use that energy from sunlight to make what? In photosynthesis, what is being made? I think photosynthesis, our photosynthesis equation was on a different week and lecture, but energy from sunlight is brought in and it's used to make a molecule that we were just talking about a few minutes ago. Glucose. So plants make their own food. The energy comes from the sun, and then they use that energy to make glucose, and then they use the energy that's in glucose to make ATP. So plants also do cellular respiration. So the difference is heterotrophs like us, we don't make our own glucose. We have to eat something to get glucose or to get other types of molecules that we could break down and use for energy. So I want to find my little ATP cycle, because when we're talking about energy, there is one figure that is so very important to me. And it's this one. And I think it's going to answer some of our other questions. So just to answer the question, Adriana, that you had, is the answer to this question sunlight or glucose? Yeah, that's, I'm not going to get you on a technicality like that, but if I ask you, what's the initial source of energy that plants get? It's sunlight, but they do break down glucose for cellular respiration. Okay, I'm going to look at this. This figure to me says a lot. And I really hope that everybody in this class can understand what's going on with this figure. This is the ATP cycle. So in our cells, ATP is very valuable. Can somebody tell me why ATP is so valuable? What do we need ATP for? Aha. Okay. So this is a challenging one, right? I think a lot of people have heard of ATP and they're like, ATP, it's energy. I know what ATP is, but ATP is just, it's a molecule, and it's a molecule that can be broken apart to release energy. So we need ATP to do work in our cells. ATP is like cash. If you want to do something that requires energy in a cell, ATP is what is used to do that. So if you need to pump something across a membrane, ATP is used, and it's used by removing a phosphate group. That's how ATP gets used. But we can't use glucose to do that kind of stuff. You can't use sugar to pump something across a cell. You can't use sugar to make your muscle contract. You have to use ATP to do that. So all the energy that's in our sugar, we have to convert that energy into making ATP so that our cells have this molecule that is readily available that we can use to do all the work that our cells need to do. In our cells, there is constantly a cycle going on where we are using ATP. And when we use ATP, what happens is one phosphate group gets removed from the molecule. It's usually a molecule with 3 phosphate groups. One of them gets taken off. And when that happens, it releases the energy. And that's how we use ATP. So that means that constantly in a cell, we're using ATP, which results in there being a phosphate group and this molecule ADP. And in order to get back to ATP from that, the phosphate group has to be put back on the ADP. And the process that is used to do that is cellular respiration. Okay, so in this image, we have cellular respiration, which is these steps that we are going to go through in lecture, going through 3 main parts and breaking glucose apart and releasing the energy in glucose and using it to make ATP. Cellular respiration is all of this stuff that's going on right here that's releasing energy. Cellular respiration releases energy, and that energy that's released is used to make ATP. And as our body needs energy, ATP is broken back down into ADP and phosphate. And that energy that is released there is used to do work, to fuel other reactions that are reactions that need an input of energy. So that's something I really want you all to focus on and think about, that ATP cycle. Does anybody have any questions? I don't know if anybody's saying anything, if I can hear you. Okay, everybody good so far?
Yeah.
And go ahead.
I was just saying I'm good so far.
Oh, thank you. There's another question, which is, what does the body do with the energy from cellular respiration? So how would you answer this one? Question number 9. Oh, I don't even see question. Here we go. Question number 9. What is the energy released during cellular respiration ultimately used for? What is that energy used to do? I'm going to go back to the slide that we were just looking at. Talking about this. In order to build ATP, energy is required, right? Where is the energy required to build ATP coming from? Not directly directly coming from food, but only when we are able to process that food through which type of process? This is really closely related to that question number 9 on the review sheet. This question says, the energy that's released during cellular respiration, what is it used for? And the answer is right here. See this little arrow that says energy from exergonic reactions? This is the energy that's released from cellular respiration right here. So all the processes of cellular respiration, they are releasing the energy that's in glucose. And that release of energy is providing the energy that's needed to make ATP. So Adriana, the answer to that question is it's used to make ATP, the energy released during cellular respiration.
Okay, thank you. That clarifies so much.
Yeah, it's used to make ATP, and it happens in steps. So what we could do now, and I don't want to jump ahead of anybody who hasn't watched all the lecture videos, but I do want to have this available to people. And I noticed that your other question was about these main steps of cellular respiration, the starting and ending molecules. So one thing we can do right now is go through that and kind of account for these different steps of cellular respiration. And like I said, if you haven't watched those lectures yet, watch them. And then this will make a lot more sense to you. But let's go ahead and I'll show you kind of what's going on with cellular respiration here. So basically, cellular respiration is breaking down glucose, releasing the energy in the bonds of glucose, and using that energy to make ATP. But it doesn't happen so quickly like that. It is going to happen through a series of steps. And in these steps, as glucose is being broken apart, some electrons are going to be passed around, and we get to see where these electrons are going by looking at these two molecules called NAD plus and FAD. And when they have electrons and they're holding on to electrons, then they become NADH or FADH2. So we're going to talk about NADH and FADH2. And what these represent is energy carrier molecules. When they get electrons, they are at a higher energy state and they can carry electrons throughout this process. And that's part of processing the energy in glucose. So we're going to make a chart, which I've already made, but you can make a chart in your notes that has an area for us to put the steps of cellular respiration, the location of each of those steps within the cell, where is it taking place, the inputs into that step, and the outputs of that step. What is the first step of cellular respiration called? Give you a minute to draw this chart and we'll see what that first step is called. Okay, anybody step 1 cellular respiration? It is I can't hear you if you're answering. I'm going to even go back to this slide where we can see The first step of cellular respiration is glycolysis. Glycolysis is the first thing that has to take place. Glycolysis takes place in the cytoplasm, so there's no mitochondria here. You don't need the mitochondria for glycolysis to take place. So we're going to start filling in this table. Step 1 is glycolysis. It takes place in the cytoplasm. And glycolysis literally means breaking apart glucose. That's the root of those words, breaking apart a sugar. So we start with glucose, one glucose molecule. So 1C6H12O6. And that glucose gets broken apart into what? You can look at this diagram of glycolysis. What is the glucose itself being broken into? If I look at this diagram, this is a chart from lecture. There are 6 carbons. I can count them. I've got 6 green circles here. And the first step is breaking that glucose apart, and it breaks it into 2 3-carbon molecules. And those molecules are called pyruvate. So in that chart, I'm going to say, okay, so I start with 1 glucose molecule and I end up with 2 pyruvates. But I also noticed that there's some other stuff going on here. I noticed that even though I have to put 2 ATP into this process to get it going, that 4 ATP are going to result. So that's a net gain of 2 ATP. So I've got 2 ATP coming out of glycolysis. Also, I've got 2 NADH coming out of glycolysis, and that's because a couple of electrons from that glucose have been used to, have been added to NAD+, and they're now banked in NADH. So I've got some ATP, I've got some energy in the bank in the form of NADH, and I've got 2 molecules of pyruvate. And I'm going to put the 2 pyruvate as an output. I'm going to say that I got the 2 NADH. And note that in order to get those 2 NADH, we started with 2 NAD+. And I'm also going to say that even though we had 2 ATP going in, we had 4 ATP coming out. So that is equal to a net gain of 2 ATP from glycolysis. And just because I like to highlight energy, I'm going to highlight those 2 ATP. And that is how this chart gets filled in. Basically, it's just going through each step, and I'll tell you what the other steps are. I put a little row here for a step that's not one of our main 3 steps, but it's called the grooming of pyruvate. And then that leads to the next step, which is called the citric acid cycle. And then the third step is called oxidative phosphorylation. Okay, now, does anybody who's here with me right now feel like you can go through these steps and the location and the inputs and the outputs?
Yeah.
Yes?
Yeah.
Okay, good. So I'm going to ask you questions then, and I want you to give me the feedback on it. Because a little bit of me is thinking, I don't know, maybe I should let them go spend some time with this and then come back and fill this in. But as long as y'all are speaking up, I'm good. So help me out with this crazy little step that we're calling the grooming of pyruvate. I'm going to go back to these slides. On the slides, you can see about, how crazy glycolysis is, how many steps are part of this whole thing, just breaking the glucose apart. It really has to go through all these steps and all these enzymes and how you get the NADH and how you get the ATP. And then I'm going to slide through fermentation right now. But basically what's happening is we're looking at, I love this figure that is on slide number 32, if you're looking in the slide deck. This figure gives you an overview of these stages of cellular respiration and how things are moving from one to another. So we've got glycolysis. Glycolysis is out in the cytoplasm. Start with glucose, end with pyruvate. Then you have this grooming of pyruvate that's happening as the pyruvate enters the mitochondria, and it's going to move into the matrix of the mitochondria, and that's where the citric acid cycle takes place, which is the next step of cellular respiration. And then it's going to move into the membrane of the mitochondria, the inner membrane, and that is where the third step, oxidative phosphorylation, takes place. So you've got this kind of overview, and then you're going to look at the steps within that. So as it's moving into as the pyruvate is moving into the mitochondria from the cytoplasm, it gets groomed. And essentially what that means is, first of all, a carbon is going to be stripped away from the pyruvate. So now it's only a 2-carbon molecule. And when that happens, carbon dioxide is produced. So I'm going to go to that chart and put in that carbon dioxide. And I'm also going to make a note that this pyruvate is converted to a molecule that's called acetyl-CoA, or acetyl coenzyme A. It got a carbon removed. It got a little tag called a CoA tag. That tag is going to help it move into the Krebs cycle. And I'm going to put this all in my chart. So this is in the mitochondria. And those 2 pyruvate that came out of glycolysis are getting converted to 2-acetyl-CoA molecules. Also, we've got carbon dioxide being produced, and it's going to be 2 CO2, because I'm not going to do the little subscripts on that. I'm not going to go and correct that. But it's 2 CO2 because you get 1 carbon dioxide for every pyruvate that went into this grooming step. And we had 2 pyruvates. So we've got 2 CO2 coming out. And then we've also got, did you see what was going on here? A little bit of banking of energy. So we've got energy banked in NADH. So that just means the NAD plus is picking up a couple of electrons, and those electrons are going to get held until the third step, or the third main step. So in addition to that 2 molecules of carbon dioxide that are produced, we get 2 NADH that were produced from 2 NAD plus that grabbed onto electrons and are now NADH. Okay, Where's the next step? The citric acid cycle. Where does it take place?
Mitochondrial matrix.
In the matrix of the mitochondria. Good. Or the mitochondrial matrix. And what's going to enter that citric acid cycle?
The acetyl-CoAs.
Those acetyl-CoAs that were just produced. So we take our 2 acetyl-CoA and essentially in the citric acid cycle, those carbons, now there are only 2 carbons for each acetyl-CoA, but those carbons are just going to be stripped apart. They're going to be stripped away and they're going to get converted into what?
Carbon dioxide.
Into carbon dioxide. Good. And that's going to end up being 2 carbon dioxide per acetyl-CoA, and that'll end up being 4 carbon dioxide total produced in the citric acid cycle. All right. What else is going on in the citric acid cycle? Let's go look at the citric acid cycle in our slides. This is a nice little slide right here. This is slide number 35. It just kind of shows you the overview of the citric acid cycle. So we have our acetyl-CoA coming in, the coenzyme A is removed, carbon dioxide is produced, but also a lot of electrons are going from that acetyl-CoA to our electron carrier molecules. So they're going to NADH. We've got 3 NADH per every acetyl-CoA. And we also have this other electron carrier molecule, FADH2. And we're going to get 1 of those for each acetyl-CoA. So since we had 2 acetyl-CoA coming in, how many NADH do we get?
6.
6. Good. And how many FADH2?
2.
2. So we can put those in here. We had 6 NAD plus that are being converted into the carrier molecule NADH. And we had 2 FAD that are now going to be this energized carrier molecule of FADH. 2. Okay, so a lot of energy in the bank, but also a little bit of ATP production, even in the citric acid cycle here. So if we look back, we could see we also get 1 ATP produced. So there's a little bit of energy that's released right here that's used to add a phosphate group to ADP and make ATP. 1 per acetyl-CoA, so that's going to be 2 total. I'm going to highlight it. How many ATP do we have total right now for our whole process of cellular respiration? Not too many, right? 4. We only have 4 right now, but we need to get somewhere close to 32 or more ATP. So obviously this last step, we're going to make a whole lot of ATP. This last step is called oxidative phosphorylation. Where does oxidative phosphorylation take place?
The inner membrane of the mitochondria.
In the inner membrane, in those folds. We call those folds the cristae of the inner membrane. This is actually taking place inside a membrane, and that's cool because you can get everything lined up in a membrane, and it has to be lined up just right to do this process of passing electrons, the electron transport chain. Where are these electrons going to come from? Okay, let me go back to a slide, and then we'll do some accounting here. Just going back to this slide, I'm moving into this image of oxidative phosphorylation, which is on slide 40. And this is a really cool process. We'll get into electron transport chains more when we study photosynthesis. But essentially, in this very last stage of cellular respiration, we have in our cells a lot of energy stored in NADH and FADH2. And those are coming from those first steps of the process. They are all ending up here in the matrix of the mitochondria. And at the surface of this inner membrane, those energy carrier molecules are going to release their electrons. So essentially, they get their electrons stripped off of them and they go back to NAD plus or FAD. So in doing that, in giving up their electrons, the electrons get passed through this electron transport chain. They go from one molecule to another, to another, to another. Ultimately, at the end, oxygen is present, and oxygen is the ultimate electron acceptor. It grabs onto those electrons, and that process converts that oxygen plus a couple of hydrogen ions into water. But what's significant about this whole electron transport chain is that through this electron transport chain, energy is released. And through the electron transport chain, that energy that's released is used to make a lot of ATP. It's made through a process called chemiosmosis, which you do not need to know for this exam, but you will need to know it for the second exam when we talk about photosynthesis. But right now, you just need to know that these electrons are given up. The electrons pass through an electron transport chain that releases energy, and that energy that's released is used to make ATP. And usually you can look at our exchange rates that happen here. Usually, if you take the electrons from one NADH, you can get about 2.5 ATP, and you can get about 1.5 ATP from one FADH2. So taking everything that we had, which is 10 NADH and 2 FADH2, we can make about 28 ATP in this last step. And I'm going to go back to our chart, and we're going to fill this chart in. We're going to say, okay, I'm looking at these energy carrier molecules. I'm even going to color those things orange because I like to keep track of things. I've got my 10 NADH from those earlier steps. They're going to give up their electrons, and that's going to convert them back to NAD plus. I've got my 2 FADH2. They're going to give up their electrons, and that's going to convert them back to FAD. All of this is happening in the electron transport chain, and it's going to release enough energy to give off about, or to make about 28 ATP. Woo-hoo-hoo, that's exciting. No, don't do that. How do I, there we go. Don't want to get my colors messed up. I like my ATP to be yellow in all of this. And then the only thing that we need to also think about here is that there was oxygen. Remember that there was oxygen that was used, and that oxygen was combined with some hydrogen ions, and that was ultimately used to make water. So I'm going to simplify this by putting our 6O2 and then our 6H2O over there. And that's how at the end of all of this, we end up with 28 plus 2 plus 2 32 or sometimes even more ATP, depending on how efficient that third step is. That's how we start with glucose, but end up with 6 molecules of carbon dioxide. We start with water, but end up with 6 molecules. We start with oxygen, but end up with 6 molecules of water. You should be able to account for all of those different parts of the cellular respiration equation and all the energy that gets transferred. And this chart just gives it to you in a little snapshot. So how did it go, Adriana? What do you think?
Good.
Does that make sense?
One thing that like super confuses me is I was like trying to follow hydrogens, but they're kind of coming out of nowhere
Yeah.
for the NADH and the FADH too.
You are so right. So those hydrogens, you're trying to follow the hydrogens, and the way that you follow hydrogens is just every electron is going with a hydrogen. So hydrogens are being pulled over, and you can see that you've got this NAD plus, and it's actually getting 2 electrons and 1 hydrogen. And that's how it's going from being positive to being neutral, because it's getting 1 hydrogen atom, which carries an electron, but it's also getting another electron. But typically you're looking at where these hydrogens are going, and that's kind of showing you where the electrons are going as well. And one of the things that you might notice if you look closely at this is that glucose is being broken apart, and carbon dioxide is being produced, but where are the hydrogens going? Because there are hydrogens that are part of that glucose molecule, and those hydrogens during all these steps in the process are going with the electrons to NAD and FAD as well.
Okay.
And it's at the very end there that you get some hydrogens back. And that's because there are free floating hydrogen ions. They're just called protons, and they're available, and they're in the matrix, and they're a big part of this whole chemiosmosis story that we're going to tell a little bit later.
Okay. Thank you.
You're welcome. Okay, folks. Well, I think that you have lots to think about, and essentially by just getting through this and making that chart together, I'm feeling like you have all of the material that you need to study for this exam. So I will have some time next week that you can check in if you want. On Tuesday, I'll send an announcement about that. But for the time being, just make sure you watch all those lectures and see if you can do this accounting yourself and go back and make sense of what's going on with all this transfer of energy. Thanks, y'all.
Thank you.
Thank you so much. See
Thank
you
you.
next time.
Is there any way I can ask a question?
Just wait just 1 second. I'm going to, okay.